All right, guys. So let's talk about concept two. This is sections 4.5 to 4.7 in chapter 4, unit 2. So make sure you read those sections because you will have a concept check that will come from those sections in the workbook. All right, so concept 2 is continuing with the cell organelles. We stopped off with the endomembrane system last time. Remember, the endomembrane system is the system of membranes that is the organelles that contain membranes and is continuous with the nuclear membrane. So it includes the nuclear membrane, the ER, the Golgi apparatus, vacuoles, lysosomes, any transport vesicles, and the plasma membrane. Now we're moving on to are double membrane bound organelles, which are the mitochondria and the chloroplast. The mitochondria and the chloroplast are unique in that one, like I said, they have a double membrane, with the second membrane of the mitochondria being there to increase the surface area. When we talk about the surface area to volume ratio, that second membrane is folded inside of the mitochondria to create these little like these areas that we call cristae, which is extremely important to oxidative phosphorylation. We'll talk about oxidative phosphorylation when we get into cellular respiration. The chloroplast also has a double membrane. It has an inner membrane and an outer membrane as part of it. The mitochondria has the outer membrane and the inner membrane as well. Another thing that's unique about the mitochondria and the chloroplast is that their origin is linked to what's known as the endosymbiotic theory, where it is believed that these organelles were formed through an endosymbiotic relationship. That's a relationship where two different organelles are working together between first an ancient prokaryote and a bacterium, and then an ancient, like an ancestral eukaryotic cell, where your mitochondria would have formed first, and then cells that already contained mitochondria would have engulfed a cyanobacterium that began a endosymbiotic relationship in chloroplast form second, which is why you can only find mitochondria in animal cells, fungus. I mean, you can find mitochondria in animal cells, but you can only find chloroplast in the plant cells, and they also have mitochondria. So the cells that led to animal cells, fungus cells, would have branched off prior to the cells engulfing the cyanobacteria. And then once that happened, those cells would have branched off into the plant cells. So again, mitochondria is found in both animal and plant cells, but chloroplast is specific to plant cells. It's one of the organelles that is only found in plant cells. Mitochondria and chloroplast change energy from one form to another. Our chloroplast is going to be able to take light energy or photon energy and transform that into the stored energy in the bonds of carbohydrates, our main carbohydrate being glucose. And then mitochondria is able to extract the energy from those bonds. And remember, the energy is in the bonds, not the elements, but the bonds. It's able to extract the energy from the bonds and then transform it to a stored chemical energy in the bonds between phosphate groups in ATP. Mitochondria is the site of cellular respiration, and chloroplasts are the sites of photosynthesis. So if we look at mitochondria, again, it is a double membrane enclosed organelle. You have your outer membrane, you have your inner membrane. And inside of that inner membrane is where it's called the matrix of the mitochondria. And this is where you're going to find your DNA. Now, the unique thing about the DNA found in both mitochondria and chloroplast is that unlike the DNA in the rest of the cell, this is a circular DNA. It's a circular chromosome. Just like that we find in prokaryotes. Another unique thing about DNA and chloroplasts are that they have their own, in addition to having their own DNA, they have their own ribosomes. Now, there's two different size ribosomes that we know of at this time. There's the ribosomes found in eukaryotic cells and the ribosomes that are found in prokaryotic cells. Interestingly enough, the ribosomes found inside of mitochondria and chloroplasts are the same size ribosomes that you find in prokaryotic cells. So they have their own DNA. There is a double membrane, which is like a cell in a cell, and it has ribosomes that are found that are the same size as the ribosomes found in prokaryotes, even though the mitochondria and chloroplasts are only found in eukaryotic cells. The matrix of the mitochondria is where your Krebs cycle is going to occur, and the cristae or the folds in the inner membrane is again extremely important for the oxidative phosphorylation process and electron transport, which leads to the production of ATP. Another unique thing about the mitochondria and chloroplast is that they reproduce independently of the cell. Mitochondria reproduces and chloroplast reproduces through binary fission. So one cell splits to become two. I love biology because it is the only place where you actually divide to multiply. Did you get it? All right. Anyway, so unlike the rest of the organelles and cells that have to go through the cell cycle, mitochondria is going to divide independently of the cell. So the mitochondria can actually undergo division even when the cell is not in the cell cycle. And it does so through binary fission, which is the same way that bacteria divides. I know I've talked a lot about chloroplasts and mitochondria at the same time. That's because they have a lot in common. Here's the structure of the chloroplast. Again, chloroplast is responsible for photosynthesis. There's that double membrane. It has its own DNA and ribosomes, which you're going to find in the stroma, which is the liquid that is inside of the chloroplast. Another thing that chloroplast has is that they have these little stacks of thylakoids, and these stacks of thylakoids are the sites of the light-dependent reactions. The light-independent reactions are going to happen in the stroma, the fluid, but the light-dependent reactions happen right here in these little stacks of thylakoids, and when you put a stack together, you call it a granum. But again, the important thing at this point is that it has its own DNA, which you see here. The DNA, the chromosomes are going to be circular like those that you find in bacteria, has its own ribosomes, which are the same size as those that you find in bacteria, and it reproduces independently of the cell through binary fission. Again, mitochondria and chloroplasts have a lot in common with that double membrane, its own DNA, own ribosomes, and the ability to reproduce independently of the cell. So we've talked about these ribosomes and chloroplasts, and again, that the things that we've talked about support the endosymbiont theory. We'll get more in detail about the endosymbiotic theory later on, but the three levels of support that you need to know about, the three evidences to support this theory, is that both organelles have that double membrane. They both have their own ribosomes and a circular DNA molecules, and they both reproduce independently of the cell via binary fission. All right, some other organelles that we're going to find in the cell. Well, another organelle that is responsible for various metabolic functions is going to be peroxisomes. Now, the origin of peroxisomes is not yet known. All we know is that it involves the transfer of hydrogen compounds to oxygen, which produces hydrogen peroxide, H2O2. This is important for breaking down fatty acids that are going to be sent to the mitochondria for fuel. This is important in detoxifying alcohols. Remember, alcohols are going to have those hydroxide groups on them. And so it's able to transfer the hydrogen from the poison to oxygen. So it's able to take those hydrogens off of those hydroxide groups and then transfer them to oxygen again to make the peroxide. This is another great example of compartmentalization in the cell. Remember, compartmentalization is the ability of an organ, is what happens when an organelle has its own membrane around it. So whatever it does stays isolated inside of that membrane so that it does not contaminate or cause any damage to the rest of the organelles or other processes that's going on in that cell. That's why most organelles are going to, in the eukaryotic cell, are going to be membrane-bound organelles. Of course, the exception to that are the ribosomes. Compartmentalization is going to keep the enzymes that produce the hydrogen peroxide and those that dispose of these toxic compounds separate from the other cellular components. Again, this is to prevent damage inside the cell or damage to other organelles. So this is a great example, along with lysosomes. This is a great example of why compartmentalization is important in eukaryotic cells. The next the next network is called the cytoskeleton network. It's a network of fibers that organize structures and activities in the cell, and it's responsible for support, motility, and regulation of some biochemical activities, like they're extremely important in the cell cycle. There's three types of fibers, and they're named based off of their size. You have microtubules. If you think about it, that's a tube. Tubes are usually bigger. And your next two are filaments. You have intermediate filaments. We know intermediate means like the middle. And then you have microfilaments. So between the microtubules and the microfilaments, remember, a tube is bigger than a filament. If you think about a filament, think about the little thing that's inside of like a light bulb. Think about how small that would be as compared to a tube, okay? Now, we're on a micro scale, so they're not that big, but that's kind of just to give you a way to remember the difference between microtubules and microfilaments. Your microtubules, again, are going to be your largest. They're part of the cytoskeleton, and they are made of a protein called tubulin. The main function of a microtubule is shape and support of the cell. So you're going to have your little motor proteins, and I need to show you guys the video of the inner life of a cell. It's so cool. But the little tracks that the motor proteins can actually move on. These are the fibers that are important in the cell cycle because they're going to form that mitotic spindle. And the mitotic spindle, the microtubules that come from the mitotic spindle, those kinetochore and non-kinetochore microtubules are responsible for moving the chromosomes around and also elongating the cell during the cell cycle. And they also play an important role in the structural components of cilia and flagella, which are the organelles that are used for movement. They have a 9+2 arrangement in both cilia and flagella, and I think I'll get to that later on. But what that basically means is, like, if you look in the inside of a cilia or a flagellum, you're going to see 9 pairs of microtubules around the outside and then 2 microtubules in the center. And the way that they interact with each other controls the movement of the cilia and the flagella. Again, I'm supposed to talk about that later, but y'all don't know. Well, you'll learn how I do. All right. Our next set of not microtubules. Next important, the intermediate filaments. I'm sorry, it's morning. Forgive me. All right, your intermediate filaments are going to be your more permanent fixtures in the cell, and your intermediate filaments are the ones that's responsible for keeping the cell shaped. So the reason why a neuron is shaped like a neuron and a fat cell is shaped like a fat cell, the reason why each one of these cells have different shapes is because of the workings of the intermediate filaments. In addition to help maintaining the cell shape, they also are going to be the ones that anchor certain organelles in the cytoplasm of the cell. So the nucleus is anchored, and your ER and things like that are going to be anchored into certain positions, which is why we need transport vesicles to get certain things from one organelle to the other one, because the other organelles are going to be anchored in that cell. And the anchoring is due to the intermediate filaments, which brings us to the last type, is on the microfilaments. These, again, are your smallest. They are going to act on a more small scale and support and movement. They basically are going to be made up of actin filaments, and you see these working in like muscle movement when the actin and myosin filaments slide over each other to contract and expand your muscles. They're they're also a part of the motor proteins. Yeah, that's it. All right. Now, our next organelle, the centrosome. Sorry, I'm trying to see if I had any more coffee. Not supposed to have coffee. That's why it's so good. All right. So the next organelle is your centrosome. Now, a lot of people confuse centrosomes with centrioles, okay? Your centrosomes are part of the regions located near the nucleus, and they're the organelles that the microtubules that are going to play a part in cell division come from. So your centrosomes are near the nucleus. They is a pair. They come in a pair and they separate during cell division to go to either end of the cell. And there they are where the microtubules originate from that are going to participate in moving the chromosomes and elongating the cell. The ones that move the chromosomes are called kinetochore microtubules, and the ones that help to elongate the cell are called non-kinetochore microtubules. But that's the role of a centrosome. Now, a centriole are actually located inside of centrosomes that are found in animal cells. So, plant and animal cells will have a centrosome, but only animal cells have a centriole. Okay? Make sure you don't get those two confused. Centrosomes, plant and animal. Centrioles are found in the inside of centrosomes of animal cells only. Nearly all eukaryotic flagella and cilia are arranged again in that 9+2 pattern that you can see here. I was talking about earlier. You probably understand it better now that you see it. There's 9 pair of microtubules around the outside. You have your radial spokes, and then you have your central microtubule, and there's 2 there. So this is what it means by 9+2. Cilia and flagella are organelles that are used for movement, and the difference is the length and the way that they move. Your flagella are usually longer, few in number. Like if you think of the human sperm cell, there's only 1 flagella. Some bacteria will have 2 flagella, but they're usually longer and few in number, and they move like a snake. I don't know if you can see this or not because I can't see myself today on here. I don't know what I look like. Cilia, on the other hand, are going to be shorter and more numerous. They kind of look like little hairs, and they move like windshield wipers. I'm sorry. Cilia, when they're used for locomotion, they're going to move like, a sweep, kind of like rowing a boat. But then they also can be used to move fluid over the surface of a tissue when they are a part of that tissue. You also have cilia in your lungs, not here, your lungs or whatever, and it helps to like sweep the stuff out. Yeah. Okay. Okay. Your other extracellular components. Extracellular means that these are components that are going to be outside of the plasma membrane. And so when you think of that, you need to think of like a cell wall and the extracellular matrix. Okay, cell wall and extracellular matrix. And there's also connections between cells, and your extracellular components and your connections between these cells are going to help to coordinate cellular activities. Now, every kingdom of cells, with the exception of animal cells, have organisms that are either all or some. So some or all organisms in that kingdom have a cell wall. Plants have cell walls made of cellulose. Fungi have cell walls made of chitin. Some protists are going to have cell walls, and the ones that do, those cell walls are made of pectin. Eubacteria are going to have cell walls made of peptidoglycan, and then your archaeobacteria have cell walls, but those cell walls are made of a combination of uncommon lipids, and that becomes the cell walls of the bacteria become extremely important when we're trying to figure out the difference between gram-negative and gram-positive bacteria, which again we will talk about later on. The extracellular matrix is what you're going to find in animal cells. Since animal cells don't have a cell wall, and we know that the cell wall is going to be important in maintaining the shape of that cell, so it's important in structure. Animal cells don't have a cell wall, so instead they have what's known as an extracellular matrix. And the extracellular matrix is going to be right outside of your cells. It's composed of glycoproteins, such as things such as collagen, and there are components like glycoproteins, glycolipids. Glyco meaning there's a carbohydrate attached to the protein. Uh, Glycolipid means there's a carbohydrate attached to the lipid. But these things are going to be a part of the extracellular matrix as well as embedded in the plasma membrane of the cell, and then they interact with each other. And so the extracellular matrix is the outside component that's helping to maintain the shape of the cell. So on the inside, you're going to have your intermediate filaments, which is basically holding up if you think of it like a wall, so from the inside, the intermediate filaments are holding up that one side of your phospholipid bilayer. Then on the outside is being held by the extracellular matrix. So you have an internal and an external force that is helping to maintain the shape of that cell. Your extracellular matrix is important because it greatly strengthens tissues and it helps to transmit external stimuli between cells, which is good in gene and biochemical regulation. Now, your intercellular junctions, okay? Intercellular junctions are the way that cells communicate with each other. In plants, that intercellular junction is a plasmodesmata. Now, a plasmodesmata is kind of like if this is a plant cell and this is a plant cell, if there is a gap in between both of the cell walls, so it's like a little hole in the cell wall where they can send messages from one cell to the other one. Internal messages can go from one plant cell to the other plant cell through the cell wall. That little hole or that little gap in that cell wall is called a plasmodesmata. Plasmodesmata are specific to plant cells. Animal cells have a similar way of communicating through a gap, but it's called a gap junction in animal cells. Okay, so animal cells through the cell membrane can have a gap from one cell to the next one, but it's called a gap junction. If it's in a plant cell, it's called a plasmodesmata. Animal cells also have two more types of intercellular junctions, one being a tight junction, which is a fusion of two neighboring cells where they're actually going to be fused together, and then desmosomes, which actually fasten those cells together in like sheets. All right, that's it, guys. Next time I'll be talking about concept check 3, which is a whole different chapter. So it'll be all of chapter 4. And I hope you understood that. If not, hit me up with some questions. Peace. Stop it.